This morning, I have the privilege of introducing Scott Minnich, Associate Professor of Microbiology from the University of Idaho. Professor Minnich received his Ph.D. from Iowa State University in microbiology, and after doing some postdoc positions at Purdue and Princeton and a teaching position at Tulane, he finally accepted his present position at the University of Idaho. Interesting little note, he also has worked on developing, in a work position that he had, some rapid diagnostic procedures for infectious agents. And I think that's part of what's led him into being asked to go to Iraq next year as part of an inspection team. So we're lucky we still got him. He's running back and forth across the country right now, but they allowed him to come here. So today, Scott will take a critical look at an irreducible complexity in nature, and in particular the E. coli bacterium, which turns out to be an incredible work of creation. He will also show the difficulties of trying to fit the development of such an organism into a classical Darwinian evolutionary scheme. These ideas will be explored further in a panel discussion following his talk, led by Art Batson. Let us welcome Professor Minnich.
Thanks, John. Well, thank you for the invitation to come and speak at this symposium. It's a real honor. I believe that, if you look at the hierarchy of the sciences, you have to put philosophy on the top, physics and mathematics next, and then biology and chemistry follow in the wake. We are dependent upon the physicists to direct us and provide us the technology that we need. But anyway, I want to talk about the bacterial flagellum. Part of this will be very specific, more than you want to know, but just to give you an idea that this is an active area of research I've been involved in, a little bit with E. coli, more with a relative in the genus Yersinia, but it's essentially the same system. I think it's important because the bacterial flagellum has been kind of the mascot of the intelligent design community in terms of looking at irreducible complexity. And there is another system that has some similarities to the flagellum. We call them type 3 secretion systems that I also work actively on. And when this similarity was recognized, people like Ken Miller at Brown said, well, here's your intermediate, here's how you can explain irreducible complexity from a Darwinian standpoint. And Ken doesn't work on these systems. I think it poses more problems for a Darwinian position, and I'll give you my thoughts on that. But I want to start first by waxing more philosophical. I'm in the biological sciences, and the position that I take is more or less heretical. We don't talk about God when we give lectures. That's streng verboten. But at the same time, I think it's important that we look at the issues and not forget the heritage of where we're coming from. But I have a couple quotes that, if you're a student in the biological sciences, you're going to run into. And they can have, cause you to pause and think about it. Now, the first one's from Jim Watson, Nobel laureate on structure of DNA. We're celebrating the 50th anniversary this year. In his first edition of the Molecular Biology of the Gene, which has gone through numerous editions, was used as a standard textbook. I used it as an undergraduate student. We used it as a graduate student, and I even used it when I was teaching in the late 80s. But in the first chapter, really on the first page, he makes the statement, today evolution is an accepted fact for everyone but a fundamentalist minority whose objections are based not on reasoning, but on doctrinaire adherence to religious principles. Okay, and then many of us are familiar with Richard Dawkins from Great Britain in his book, The Blind Watchmaker. he starts out, although atheism might have been logically tenable before Darwin, Darwin made it possible to be an intellectually fulfilled atheist. Okay, and then whenever these debates come up in the school systems in Kansas and Ohio, you will see Dobzhansky's quote bantered about, the last one, "Nothing in biology makes sense except in the light of evolution." All right, these are pretty strong blanket statements. But I think it's important to recognize that all biologists agree that the things that we study have the appearance of design. Dawkins will admit this as well. That's the whole point of writing The Blind Watchmaker, was to try to convince his readers that it's only apparent design, it's not real design, that the mechanism of mutation and natural selection is sufficient to explain the diversity of life that we see on this planet. But the question is there. This has been an old question, from recorded history. Materialists and theists arguing back and forth. Is what we see in nature the product of a higher intelligence, or is it merely chance and necessity, as Jacques Monod would want us to believe? The problem that we have in the biological sciences is that we have, I think, in our triumphal arrogance, in the success and the changes that we've had just due to experimental or empirical science, have drawn a conclusion that may be a little bit premature. Okay, so my point then is, we've got to, I think, always go back to these questions and make sure that our arguments are buttressed. Science is not the purely objective endeavor that often we're led to believe. It's very much personality and worldviews that conflict. This first one is from Murray Gell-Mann, a physicist that in 1979 wrote, "The fact that an adequate philosophical understanding of quantum mechanics has been so long delayed is not doubt caused by the fact that Niels Bohr brainwashed a whole generation of theorists into thinking the job was done 50 years ago." Okay? Jacques Monod, when he discovered the first genetic regulatory system and the repressor involved in the lac operon, assumed that all regulation was by negative transcriptional control. And those people that were beginning to have evidence that there was positive transcriptional control involved in these systems were, by the force of his personality, not allowed onto the field until the evidence was overwhelming. So you go back to the original statements that I had up there, which are essentially saying, hey, the game's over. We won. Everybody can go home. I think we have to reevaluate. Here's a quote from James Shapiro, University of Chicago. Jim Shapiro is the individual that in part discovered transposition elements in bacteria that shed light on Barbara McClintock's work done in maize years before, in our whole idea of jumping genes or transposons. And several years ago wrote in one of our publications, "There are no detailed Darwinian accounts for the evolution of any fundamental biochemical or cellular system, only a variety of wishful speculations." And this is, by and large, I think, as we look at these as the examples, whether it's antibiotic resistance or peppered moths, microevolutionary examples, we find that as they're put under the microscope and examined closely, that oftentimes it boils down to just so story that Rudyard Kipling would have penned. It's the question over. Now this is the introduction of a paper that was published last May by Lenski, who's at Michigan State. Robert Pinnock was a co-author on this paper. He's a philosopher of science. He's published a book by MIT Press looking at the neo-creationists, and I think his book is titled The Tower of Babel. But I want to read this and consider what they're saying. From the outset, now this is a paper in Nature, this is a scientific paper, and I'll give you the context of this statement as we continue. But at the outset, Darwin realized that origins of extreme perfection and complications, such as the eye, posed a difficulty for his theory. Such features are much too complex to appear de novo, and he reasoned that they must evolve by incremental transitions through many intermediate states, sometimes undergoing changes in function. All right, so you look at the eye, this was Paley's argument or the watchmaker's argument. How do you get something so complex? Well, it's got to go through incremental changes. But then they further state, now there exists substantial evidence concerning the evolution of complex features that supports Darwin's general model. Nonetheless, it is difficult to provide a complete account of the origin of any complex feature owing to the extinction of intermediate forms, imperfection of the fossil record, and incomplete knowledge of the genetic and developmental mechanisms that produce such features. All right, now, as a member of the intelligent design community, we're accused of being pseudoscientists, that our approach is unfalsifiable, that it's not scientific. Yet, here's the question. We've got these complex systems. Are they designed, or can the laws of nature, physics and chemistry, produce these unaided by any form of intelligence? And they're going to perform scientific experiments to verify this fact. We don't have the intermediate forms. We don't have the fossil record, and we don't have the mechanism, genetic, biochemical, to explain how you get from a simple system to a more complex. All right? Now, Lenski has been doing experiments over the last 20 years in continuous fermentation apparatuses, looking at E. coli, looking at Saccharomyces cerevisiae, running them under constant conditions in fermenters out to 20,000 generations, and putting them under stress and looking at the changes that the organisms acquire, and they do adapt to their environment. But when you're done after 20,000 generations, you still have E. coli, you still have Saccharomyces cerevisiae. You haven't generated anything complex. In fact, the conclusion from a paper that was published by Lenski in April of the same year, more or less said it's surprising how little change we do see when we run these experiments. So in this paper, what they did was generate a computer program, create virtual organisms, allow them to compete, and see if they could develop more complex lines of code. And they got what they wanted in 15,000 generations. Okay. So you'd run the real experiment, you don't get the result you want. So then you can set up a virtual experiment on a computer, and lo and behold, you get the complexity that you want. I think it's a shell game in terms of where the intelligent input is. All right? But seriously, they're using scientific evidence to dismiss the argument of design. And yet, if I'm going to engage in this conversation, my hands are tied, because if I infer an intelligence or supernatural, that's out of the realm of science, and therefore out of bounds. Okay, it's not a fair playing field. I want to point out too to many of you, perhaps you're unaware, that why have we had such success in the sciences in a Western culture that had as its worldview or foundation a Judeo-Christian point of view? I think this is accepted by secular historians, and I think Nancy may address this later this afternoon. In fact, there's a new book that we were talking about last night. But this is a quote from Francois Jacob's book. Now, he was Jacques Monod's cohort. They shared the Nobel Prize for their work on genetic regulation back in the 60s. He wrote a book titled Of Flies, Mice, and Men that was published by Harvard Press a few years ago. He's still alive. And he states, between the 13th century and the classical age, Europe transformed its entire culture. Not only was symbolism replaced by representation, but you had this revolution in music, prayer by action, mystery by drama, the narrative by the novel, the chronicle by history, and the myth replaced by scientific theory. Nor should we forget that the structure of the Judeo-Christian myth was largely responsible for the development of modern science. The former was founded on the doctrine of a prevailing order in a universe created by a God who was himself not part of nature, but who directed it by means of laws intelligible to human reason. So this goes back to this question of a transcendent God or creator that Michael talked about last night. They recognize this. God is outside of the system that we live in, yet the system is directed by laws that are intelligible to human nature. Going right back to Romans 1, God has revealed himself in what he has made. We are compelled then to study nature because it tells us about the Creator. So you have the aesthetic side of the equation, as well as we are to love our brother as our fellow, our neighbor as ourself, and that through the study of nature, understanding these laws, we will develop technology and we can make life better for everyone on the planet. Okay. These were the directives that I think were the foundation or the underpinning of Western science. You don't see this coming out of other worldviews. In fact, as a professor and having graduate students from other cultures, this is one of the things that's really difficult at first to get across to students, that you go with the data, regardless of how it, meshes or conflicts with your hypothesis, you go with the data. How many times I've had a student from India that has run an experiment and contradicted what I thought the results should be, yet has told me that it worked perfectly well, just like you predicted. And so we go on and do the next experiment, and we just keep building on this house of cards until finally nothing fits, and you go back and say, let me see the data. I thought you said this experiment worked. Well, no, it didn't. It's evident that it didn't. We got the opposite answer. Well, why didn't you tell me that? Well, I was afraid I would offend you. You have this cultural barrier that, whatever the chief says, you better go along with it because you don't want to be involved in him losing face. So this is one area that, in science, you've got to go with the data, regardless of how or what you think. Freeman Dyson, the physicist at the Institute for Advanced Studies in Princeton. I enjoy the books, and he's been involved writing about origins of life. In his book, Imagined Worlds, he has this quote. The poet W.H. Auden, who was a Christian, wrote of the importance of Christianity to the birth of modern literature in late antiquity. One may like or dislike Christianity, but no one can deny that it was Christianity and the Bible which raised Western literature from the dead. A faith which held that the Son of God was born in a manger, associated himself with persons of humble station in an unimportant province, and died a slave's death, yet did this to redeem all men, rich and poor, freemen and slaves, citizens and barbarians, required a completely new way of looking at human beings. If all are children of God and equally capable of salvation, then all, irrespective of status or talent, vice or virtue, merit the serious attention of the poet, the novelist, and the historian. Auden made a strong claim for the impact of religion on our image of ourselves, an equally strong claim for the importance of science. Okay, again, this is just emphasizing that this worldview of a transcendent God that is both infinite and personal had a profound effect in terms of how we looked at nature. And we have, reaped the benefits of it. It has paid off an astonishing coin. When I engage in these conversations with my colleagues, and ask them, will you agree that nature has the appearance of design? They say, sure. Well, why are you unwilling to engage in the arguments? I get two responses always that I find very interesting. One is, I don't like the idea that there's a God out there who's paying attention to what I'm doing. Or, if there's a God, I don't want to know him because there's so much evil in the world. Okay. Steven Weinberg, you'll see the same thing in his writings, this problem of evil. And when I have these conversations, I say, fine, those are legitimate positions to take. I understand them. They're difficult questions, but recognize that you're rejecting my position not on the basis of your science, but on theological considerations. You don't like the implications. You don't like the metaphysical implications of where they lead you. I think that's important. So coming back before I launch into the specifics, we get painted with this brush that we are creationists. We want to get the Bible back into the classroom. We want to prevent the teaching of evolution. That is not the case. Okay, I have no desire to use the lectern as a pulpit. In fact, I'm very careful when I teach students that if we cross this line into philosophy or metaphysics, that I'll alert them that, hey, this is my understanding if it comes up. Robert Minnich was at the University of Idaho a year ago, gave a seminar on his new book, Tower of Babel. In his PowerPoint presentation, my mug came up and said, you know, watch out, Idahonians, right here in River City, you've got a problem. Scott Minnich is running a haven of graduate students, you know, and teaching them intelligent design. And I thought it was interesting this summer when I was at a meeting with several other faculty and their students, and some of the graduate students were rooming together. One of my graduate students was rooming with a biology graduate student, and he asked him specifically, you know, how do you deal with Minnich and these crazy ideas that he has on origins of life and evolution? And my student said, what are you talking about? You know, I work on type 3 systems. You know, I didn't know he had these thoughts. I've never paid attention to it. And so quite the contrary, you know. So he brought this up on the ride back from the meeting, and I said, okay, if you want to know what I think, I'll tell you, but recognize these are my thoughts. I'm not going to judge your performance in my lab on whether or not you agree with me. And I think that's the way things should be done. All right, I think we should teach evolution. I think we don't teach it well enough, that we're not looking at it critically, and as the data changes, as our understanding changes, then we need to address those changes. All right, a couple other quotes. Paley, Reverend Paley presented his argument of design back in the 1700s. Natural history was a strong argument for a creator in his watchmaker analogy and use of the eye. I think he extended it a little bit because at his day and age, they tended to look at creation as being perfect. And I think if you look at creation, you see that it can be very cruel. Darwin had a problem with this, and some people actually think in part his development of the theory of evolution was to remove God from the responsibility of evil in nature. Nature is extremely cruel, so evolution removes God from the responsibility of evil presented in nature. Okay, we've all seen this. Let me digress for a second. When I was an undergraduate, I grew up in large part in Northern Virginia, but went to school in Washington State University as an undergraduate. I like to backpack. I love to fish. That's why I'm in Idaho. And that's work. Being in the lab is my avocation. But we had some friends high school friends that came out to visit us, and they were getting into animism. This was in the late 70s. So we're backpacking, and every morning they get up and talk to the trees, talk to the flowers, talk to the spiders, you know. It's just, you know, I mean, oh. And we're on the Lochsaw River, and we were sitting resting in the early afternoon, and this osprey came down right in front of us and just plucked a beautiful 16-inch cutthroat trout in front of us. And our friend Dana was sitting there watching this, and I said, boy, old brother osprey just nailed brother trout. Look at that. Nature is cruel, you know. There's danger out there. Okay, so evolution, what is it? It's descent with modification. Natural selection works on mutations in a continuous fitness economy stream. Mutations occur, and over time, new species are developed, others are lost. That's the mechanism, natural selection. Evidence is based on three primary sources: observable changes, microevolution, antibiotic resistance in bacteria, color changes in peppered moths, the fossil record, and homology, genetic, biochemical, and structural. You know, so all of these kind of make up the overwhelming evidence for evolution that Lenski referred to in the introduction of his paper. yet they're all incomplete. And alone, I think, individually, we have problems. What's new? Molecular biology has generated a revolution of our understanding of the cell. The cell is run by an assemblage of machines that really tax our imagination. Bruce Alberts, the president of the National Academy, published a paper in '99 in Cell, saying that when he was a graduate student, their view of the cell was a bag of enzymes that functioned on second-order kinetics. Now we find that they're this assemblage of macromolecular machines. In fact, the whole purpose of writing this paper in Cell was saying that we're going to have to include engineering, design engineering, in our biology curricula for the next generation of students to understand how these machines talk to each other, interact, all right? So you're gonna have to be a design engineer, even if you're not willing to ascribe any of these systems as a product of a designer. And the web page for the Department of Energy, which has put a lot of money into the genome projects. And this may have been removed, I think, just because for the obvious reasons that it's a conundrum. Biological systems have achieved levels of intricacy and subtlety that dwarf the engineering feats of the 20th century. Okay, we've got nanomachines that dwarf our own engineering ability, yet we're willing to ascribe their origin to non-intelligent, unpurposed products of mutation and natural selection. This is a recurring theme in the writings of the first generation of molecular biologists that are retiring. Lucy Shapiro at Stanford had a similar paper in the Journal of Bacteriology saying, who would have guessed, you know, the complexity of the DNA replication machinery? Nobody predicted it, yet here we are. The point being that the stakes now are higher. The order of complexity is, I would say, orders of magnitude above what we thought in the 1960s and 1970s. The mechanism to generate that complexity has remained constant. We still have mutations and natural selection. And you can throw in a little bit of lateral gene transfer, but that's really shuffling the deck. And you know, you're not generating new information. I think the challenge then to neo-Darwinism, and that I'm coming up against, is then to explain how chance and necessity can build machines that outstrip our intellectual capabilities. We want to know how these machines work because we can use them. We can build computers based on the bacterial flagellum. You know, it's a on-off switch, two directions, you know, a bimodal switch. That's great. It's biological. Very fast response times. How did the blind and undirected laws of physics and chemistry come up with the most sophisticated information system we know of in the universe? Right? DNA is the most sophisticated information system we know of. It's a true code. It's just like our alphabet or our musical scale. You can mix nucleotides and get infinite noise or beautiful music and poetry. Intelligent input is the required distinction. Consider then, I mean, really, what faith it requires to consider that at some point inorganic molecules associated themselves together, formed a replicating unit, developed into a cell, developed into multicellular systems, eventually to the point where creatures like us that are an assemblage of trillions of cells could then contemplate their own existence. It's mind-boggling. So we have this problem of information. Where does information come from? And the problem of irreducible complexity that Mike Behe threw out on the table a few years ago. Is it scientific? Can we detect design? Well, Bill Dembski's been, kind of leading the charge on this for the last few years in terms of how to really define information or how you can quantify what's designed, and his whole theory of complex specified information, I think is a really important contribution, although oftentimes it's dismissed. Again, this criticism that it's unscientific. If designed, it's a science stopper. It can't be falsified. Everybody go home. If you're going to invoke the supernatural, then, hey, there's nothing left to do. Well, that's not the case. I remember listening to Paul Erdos, who was allegedly the most famous mathematician of the last century. And he had this concept of, the supreme fascist that put all this stuff together but didn't leave the blueprints in terms of how things worked. That's our job. And I can ascribe the bacterial flagellum as being, in part, the product of a supernatural intelligence, but that doesn't tell me how it works, how it assembles. And I want to know, okay? So I just look at it differently. I look at it from perhaps a design perspective. We use design in a lot of disciplines. Forensics, was this natural, or was this, crime the product of intelligence? Anthropology, the SETI project, it requires a recognition of pattern, something, this complex specified information. Before I came down, I threw these rocks on my scanner. One of them I've had since I was in first grade. It was a gift from my cousin, who found one of these rocks on the banks of the Potomac. The other I pulled off my driveway before I came down. They're all, if you had a geologist, you can analyze them in terms of I think there's similar makeup. But why would I keep one and not the others? Okay, one of them was designed. It had a purpose. You've got this one on the lower side here as an arrowhead, and a good anthropologist would probably be able to tell me when it was made, what tribe made it, and what it was used for. Okay, so that's where design comes in, detecting design. So again, we come back to this question, life as the appearance of design. There are two answers to the question. Is it real or only apparent? We should be dealing with both. Science has not shown how we can achieve the sophistication of the design that we see in nature. I think the inference to the best explanation is that there is a designer hanging out there somewhere. Okay, so let me get to the specifics then and tell you a little bit about bacterial flagellum and this other system, the type 3 secretory system that has been used as an argument against irreducible complexity. Now, I work on pathogenic Yersinia. So there are 3 species here that cause us problems, that do us harm. Yersinia enterocolitca is a foodborne disease organism. Pseudotuberculosis, also foodborne, and Yersinia pestis, which we just got a major NIH grant to look at in terms of developing vaccines, is the etiologic agent of bubonic plague. And one of the things I'll be running around the deserts of Iraq looking for. Okay, now these are first cousins to E. coli and Salmonella, and so most of what I say will apply to these other systems. Yersinia are a model system for understanding disease at the molecular level, and these are some of the contributions that we've acquired from them. They are the paradigm system for type 3 secretion, which I'll explain in a minute. So these were discovered first in Yersinia and found to be really a component in all gram-negative bacterial pathogens. So here was the observation when we started this project about 12 years ago. Yersinia enterocolitca makes flagella. These are the organelles that propel the cell at 30 degrees and represses does not express virulence proteins at 30 degrees Celsius. But at 37 degrees, this is mammalian body temperature, flagellum biosynthesis is arrested, and you turn on this protein secretory device that will actually inject toxins into your system. Okay, now, our questions were, how does temperature regulate gene expression? That was a basic question. In this very narrow temperature range of 30 and 37, and why is motility shut down in the host? This is a general theme for many bacteria that cause disease. Right, now I want to go back for a second, and I wanted to show you what the flagellum looks like. This is the surface, the outer surface of a gram-negative bacteria. This is the inner membrane. This is the flagellum. We call this a basal body or rivet structure. It's got a stator, drive shaft, bushings. This is referred to as the hook, which functions as a U-joint, and this is the propeller. So this thing is assembled sequentially from the inside out. Now when we're talking about type 3 secretion, which the flagellum is an example of, and these virulence systems, we're talking about how the cell will direct these proteins to their appropriate position. All of the components of the filament are actually directed through this hollow core out through the flagellum and assemble on the tip. So the flagellum has been referred to as the most efficient machine in the universe by Howard Berg. It self-assembles. This thing will spin in E. coli at about 17,000 RPMs, forward and reverse. Requires about 50 proteins or genes to make it, 30 structural components, water-cooled, proton motive force, a whole bit. And there are gatekeepers here that will count the number of components to build these rings. And once that's assembled, then there's feedback and saying, okay, that's done. We need the next proteins to come through. So they're secreted in an ordered fashion. If there's a mutation in any one of these systems, it feeds back then and shuts down the entire process. Costs about 10% of the total cell's energy to run these machines. They're very expensive. You can't afford to produce all of this protein if you've got a non-functional system. All right, so this will actually just show you or illustrate in an animation form the assembly of the flagellum. So these are the M ring that are assembled first, about 50 subunits. You see the drive shaft of the flag proteins coming through. Once they're assembled, you get your next ring, your bushing. Anchor this to the cell wall. You assemble your U-joint or your hook protein, the exact number required to give you about a 60-degree bend to the structure. You assemble a ratchet on the top of this, a scaffold, so when the individual filament proteins come through, they'll polymerize and form a helix, a cross-section of which is a beautiful propeller, okay, as you can see, with the blades sticking out. Okay, so there are, I think, about 20,000 subunits of the actual filament or the propeller itself that will assemble about to an order of 10 lengths of the flagellum. It has a helical pitch, and this is what then a bacterium looks like as it's swimming. Okay, these are, again, sophisticated machines. We can't build these yet, okay? And when we will do that, but it will be using what we find already in nature as a template. They're remarkable. Okay, where was I? So I'm defining the system. Let me go back to the slideshow. Our observations, the questions we wanted to ask. Now, at the time we began this, we didn't know anything about type 3 secretion systems. But here's Yersinia enterocolitca that is growing at 30 degrees Celsius. You can see these hair-like structures on the surface of the cell. These are the flagella. If we grow them at 37, they're not made. So we want to know why. Is there a reason for this? And conversely, we find activation of the weapon systems, which would be induced inside the host. These are turned on concurrent with repression of flagellum biosynthesis. Now, we thought these are two systems that were easy to study because the phenotypes or the outward expression of the cells are easy to monitor, and we just chose them arbitrarily. We didn't think there was anything actually related. But in 1990, a friend that I'd postdoced with at Princeton, when we'd worked on Caulobacter, the flagellum biosynthesis, called me up and said, what's the function of this gene here called LCRD? It's referred to as low calcium response D. And I said, well, it's on the virulence plasmid. So all of these pathogenic bacteria have a small piece of DNA of about 70,000 base pairs that's apart from the chromosome that's required to cause disease. All right, so this thing is a genetic map of a plasmid that we find in Yersinia pestis or Yersinia pseudotuberculosis or Yersinia enterocolitca. This piece of DNA has killed estimated 200 to 300 million people, all right, in the course of our history. It's an important piece of information. And for disease to occur, you have to have this gene LCRD, and when it was cloned and sequenced, it was found to be similar in predicted amino acid sequence to a flagellar protein. So David Mullen at Tulane calls me up and said, "What's this doing in Yersinia on the virulence plasma?" And I said, "Well, we don't really know, but we have mutations. It blocks secretion of these virulence proteins." All right, this was our first hint that virulence had something to do with the flagellum. In the course of the next year or so, Lucy Shapiro at Stanford, also working on Caulobacter, found that these genes that are also required for virulence contain homologs of flagellar proteins. All right, and there are about 10 of them. So now you have 10 proteins that are involved in biosynthesis of the flagellum, 10 proteins that are involved in secretion of anti-host factors, toxins, stuff that will kill you. What's the relationship? And suddenly it became a very interesting story. Now this is just a gel showing that we do gels in our lab, but this is if you take cells and grow them at 30 degrees, spin the cells out and just precipitate any proteins, you don't get anything. But if you grow them at 37 degrees and remove calcium, you induce this virulent system. And from about 3 mils of cells, this is 5 microliters that was concentrated. These are all virulence proteins. These are various toxins that will do you harm. They're secreted in abundance. In fact, they make so much that you can go in there, you actually see it coagulating in the growth tube, in the test tube. Okay, so these are referred to as YOPs for Yersinia outer proteins or secreted proteins. Okay, so we had then both of these systems. They're coordinately regulated in a reciprocal manner. 30 degrees, you make a flagellum, you don't have your weapon systems on. 37 degrees, mammalian temperature, the bacterium is using that as a cue from the environment that I'm in a pretty dangerous position. I'm going to have to deal with an immune system. You better activate your weapons, and Yersinia are very good and skilled at doing this. In fact, they actually live in our white blood cells, and they go right for the Pentagon, okay? So these are major leaguers. So they share similar proteins in this process. So the question arises, do these flagellar proteins and YOP secretion require shared components, involve separate parallel systems, or represent independent but cross-functional systems? These are all different hypotheses. Using Occam's razor, we just assumed the easiest explanation would be, let's look at the bacterial flagellum not as an organelle to propel the cell, but as a dedicated protein secretory device that's very efficient when you look at how much flagellin protein. Let's just switch it over and use it for secreting different proteins under a different environment. Why not? You would think my Darwinist friends would say, great, that's just what we predict. But when you put this in a grant and show your preliminary evidence, mutants that support it, this is a whimsical idea. There's no reason whatsoever to believe that these two systems have anything to do with each other. Go to a meeting, and I would get ridiculed by some of my friends saying, Minnich, we're so glad you come to these meetings because you have such a great imagination. So anyway, but this was the slide that we used at that time to illustrate it. 30 degrees, Yersinia make a normal flagellum, just like E. coli and Salmonella. Switch to 37, you shut off, and we showed that it immediately shut off transcription of the filament proteins, and you use the flagellum as a cannon to shoot these YOP proteins, these anti-host factors. You even have a smoke ring here to illustrate that point. Okay. Now, at the time, we thought that these proteins were actually just directed to the surface of the cell and then released, these virulence proteins. Our hypothesis said they had to come from discrete locations on the surface of the cell, that there were organelles directing this. And in fact, in 1994, we were excited when Hans Wolfwat's lab, with this paper by Rovquist, this made the cover of EMBO, it's a very prestigious journal, showed that here's a computer-enhanced image of a Yersinia pseudotuberculosis sitting on the surface of a white blood cell. So these are the actual micrographs over here. and it's stained with an antibody specific for one of these anti-host proteins. This is looking from the top down, so these are lighting up here. With confocal microscopy, you can do these virtual dissections and cut away then from the side of the cell. Here's the surface, here's our bacterium, and then as we go further in, these proteins are being injected directly into the host cell. All right, this told us that they're coming from discrete locations, consistent with our hypothesis. We were excited about this, and in fact, if you count the number of sites in which these are coming out, it's equivalent to the number of flagella these organisms would make normally at low temperature. All right, so our model had predictions. It should say that a class of motility mutant should be unable to secrete YOPs. We screw up the flagellum, we're going to also affect the ability to secrete these virulence proteins, and we got those mutants. A subset of flagellar genes must be expressed at 37 degrees. So you've still got to make this basal body secretory apparatus even though the cells can't swim, right? And Yersinia pestis, the plague organism, which is non-modal, was defined as being non-modal since it was isolated by Alexander Yersin, would have to have flagellar genes, a subset of which would be expressed. So these are all, these are, I think, sitting down with students saying, okay, how would we make this system work? How would we design this? These are possibilities. We can go into the lab and test them. And we found all of these to be true. In fact, Yersinia pestis does have the full complement of flagellar genes, but it's acquired mutations in a few of them. Here's a blueprint of how to make a flagellum, kind of, if we lay out that video in sequence. And I want to point out one thing. There are checkpoints in this process at each stage. So once you build a hook, whether you continue to make the filament, attach the filament, which is 90% of the mass of this structure, you have to make sure everything's in place. If it's not, there's a protein that actually stays in the cytoplasm that negates the function of this protein here, which is required to turn on the last step. If everything's in good shape, then this protein flag M, which is the agonist of this one, fly A, is secreted into the medium. So you get rid of your negative regulator by secreting it out into the environment. Beautiful, beautiful system. Okay. Type 3 virulent system has the same strategy. Okay. It does the same thing. It's got a negative regulator that you secrete out of the secretin structure. Here's what the type 3 secretion apparatus then looks like in a cartoon from Guy Cornelis' lab, that you build this structure on the surface. These are all of your virulence proteins then that are required. If you make contact with a macrophage, a white blood cell, these two proteins, YOP-B and D, act as Bangalore torpedoes, blow a hole in the membrane, and everybody runs through behind them. So these are the ones that have specific targets in this white blood cell to disable it so that your innate immune response is not activated. So we were predicting then that there would be a structure that looked like the flagellum. In fact, we thought it was the basal body that would do both. We actually did the experiment where we knocked out the master control gene, knocked out motility, and we still got secretion of these proteins, so we were wrong. But Jorge Galan at Yale found in salmonella that these needle-like structures, which are referred to as secretins, are required for secretion of these virulence proteins. So it's very similar in structure and size as the basal body for the flagellum. Okay, and I'm going to wrap this up here because I'm a little bit getting on the end of the time, but so again, emphasizing that all these other criteria were true, but the bottom line when we did the experiment, the final one, let's try and make our hypothesis, invalidate it, and we did, which was a bummer. This is just showing that Yersinia pestis makes, has genes for flagellum using reverse transcriptase PCR. We can detect messenger RNA from all of these different flagellar genes, so they are expressed, even though the cell is non-modal. We actually cloned and sequenced the master control switch from the plague organism, and it has a single T insertion, a mutation at this position that negates or throws, we call it a frame shift mutation, downstream. So that's why these cells are not motile, due to this single mutation. In fact, what I found interesting was then I had, with my graduate student, we sat down and pulled out all the other flagellar genes downstream. So this is the master switch, this FLHD. It's got to have this, you know, to activate the entire sequence of events. And we found at least 3 other mutations in the genes downstream. Okay, actual deletions or insertions. I bring this up because it comes to this question of irreducible complexity. This organism has all the genes required to make a flagellum. It gets a mutation in one. All the other genes then are rendered non-functional. You only have to make this one step to introduce motility back into the cell. But what's happening? You're acquiring now these other genes have no function. They've lost purpose, and they're just sinks for mutations, neutral mutations. You lose it. And bringing this up to Darwinist friends and saying, if you're going to build something by a gradual mechanism, how are you ever going to get to the final stage? If you're just missing one component, you know, this is like trying to herd ducks. Everybody else is, you know, losing function. And they said, well, that's obvious. I mean, if you don't use it, you lose it. Well, you can have it both ways. That's the point. You know, so it is irreducibly complex by definition. I'm going to skip this slide. So if we look at the flagellum in these type 3 systems, these virulent systems, they both are dedicated secretory devices. The proteins don't use the general secretory pathway that we find in bacteria. They show similar regulation. This flagellum protein that I mentioned, once secretion initiates, there's a feedback to increase or upregulate those genes. How coincidental is it that you have 2 systems that are involved in secretion of protein that use negative regulators when it's not the proper time to secrete these proteins, and you get rid of that negative regulator by secreting it out into the environment? These are different proteins, they have different targets, but they're using the same strategy. I find that tremendously coincidental, okay? Now that's what a design engineer would do. You know, you've got a system that works, just apply it in a different environment. And then we asked, okay, these systems appear to be similar. Why do we regulate them by temperature if we are wrong in the sense that the flagellum can't do double duty? These are separate parallel systems. Everything on that plasmid encodes for that structure to secrete the virulence proteins. The chromosome contains the information for the flagellum. Are they segregated by temperature? Because if they were both expressed at the same time, they would recognize each other's substrates and mix them up. So there's a possibility. And we could test this. We could take the gene for the filament protein, the flagellum, the propeller. It's repressed at 37 degrees. You don't make any messenger RNA. You don't make any protein. Put it behind a different switch. Turn it on arbitrarily. Now we can activate it at 37 degrees and ask, does it get secreted? And does it get out the virulence system? And likewise, we could do the same thing with a virulence protein that's repressed at 30 degrees. Turn it artificially on at 30, and ask, does it use the flagellum as an exit port? And we do the experiment. I won't go into the details, but that's exactly what happens. All right, so you have these 2 parallel systems that are regulated and segregated from each other because they have to be, all right? Otherwise, you'd mix up. You'd start secreting. Put it this way. We now know in the last 5 years that the filament protein of the gram-negative bacterium is a very potent, what we call cytokine inducer. Our innate immune system, nonspecific immune system, keys off the presence of bacterial flagellum. If it's around, you know, our defenses have been breached and you initiate the inflammatory response to knock these organisms out, where your specific immune system then can catch up and make antibody and neutralize the threat. it then makes sense if you're a pathogen, you want to shut down motility when you're in the host because you don't want to single it, especially Yersinia. The hallmark of Yersinia infection is there's no inflammatory response, and we think that repression of flagellum or loss, sacrifice of our motility system is, like Yersinia pestis, is essential for that process. Flagellum can get out the type 3 virulent system. These yacht proteins can use the flagellum as an exit port if they're turned on at the same time. And what's even more remarkable is these type 3 secretory systems, that plasmid actually has regulatory genes on it to make sure that if there's another type 3 system in that cell which it's harbored, that it will override it under certain conditions, make sure that it's shut down. So you've got to have this scenario. What Ken Miller would say is that the type 3 system is just a intermediate of a flagellum, and that at some point you're able to polymerize all these proteins and put a rotary engine on it and spin it. Now don't buy it, okay? You can't, because as soon as you have these two systems in the same cell, they're competing for each other's substrates, and you lose one or the other, all right? Yersinia pestis has lost its ability to make a flagellum because I think it's getting interference from its virulent system. Shigella, dysentery, also an important pathogen, is non-modal, but it's got all the genes for flagellum biosynthesis, and it's got a type 3 secretion system. It's sacrificed it. You can't operate them simultaneously. Bordetella pertussis, when you get your DPT shot, okay, whooping cough, same thing. At one point in its history, it was modal. It sacrificed motility because it acquired the ability to do you harm, and you can't operate both systems. And E. coli 0157H7, the one that's such a problem in hamburger, in collaboration with Peter Fang at the FDA, we would then predict that if you lose motility, you might heighten virulence, and sure enough, the isolates that are coming out of Germany and northern Italy of this strain of E. coli are non-modal, and they have a 12 base pair deletion in the same gene that Yersinia pestis does, in the same gene that Shigella does, in the same gene that Yersinia does. Okay, so I think this is predictive, and what I'm saying is that I'm using a design paradigm. How would I make this system? How would I get it to function? Predicting that these systems are going to be similar. And my colleagues from a Darwinist viewpoint are saying this is whimsical. You're out to lunch. But as soon as the structure's isolated in the ciliary scene, exactly what Darwin would have predicted. Isn't that convenient? Darwinism is postscriptive, okay? The interpretation is always put on after the fact. I don't know, when I ask my colleagues, what principle of evolution do you use to design your experiments? They can't give me an example. And I say, well, some of our graduate students obviously use the principles of Darwinism. They just go in there and randomly mix things together. And, but, there are results. So this is kind of a cartoon then, that these things are segregated, that, Yersinia has type 3 systems, but you can't run them at the same time. It's detrimental to the cell. It's not what you would predict. You'd have to go through an intermediate stage where these things are compatible. And I think before that ever occurred, you would lose one system or the other. And furthermore, we now know from the phylogenetic analysis that the flagellum came first before these type 3 systems. They're more conserved. So this was also, the type 3 system is simpler. It's not an intermediate. It's a, if anything, a degenerate form of the flagellum. It's not something that upon which was built more complexity. Okay, so the summary then is essentially that I don't think this type 3 secretory system is an explanation to discount irreducible complexity in the flagellum. These systems are highly complex. They were not anticipated nor predicted. That we used reverse engineering in the laboratory to determine the function of these systems, that this is a design process. And the spin that we put on it in terms of the interpretation afterwards, from a Darwinian standpoint, is postscriptive, not predictive. All of, nothing in biology makes sense outside the light of evolution. I raised that actually Minnich leveled that at me. How could I be teaching things that are counter to the very foundation of my discipline, and really accusing me of being a heretic in front of my own students. And I said, well, that's interesting, because in my entire career, I've never once taken a formal course in evolution, have been required to. My department at the University of Idaho is the most successful in terms of publication and funding. We don't have a course in evolution as a requirement, nor is it even listed as an elective. You can do biology fine without understanding Darwinism, because when you get to the bench, you're using this idea that the supreme fascists didn't give us the blueprints for these things. You got to break them. It's like a kid in a garage, with an old car. Take it apart and put it back together and figure out how it works. And that's very much an engineering process. So I'm going to stop there, and I'd be happy to entertain any of your questions. Welcome them. Thanks for your attention.
My question is maybe the theological implication of virulent, I don't even know science and all that stuff, but I know a little bit of what you're talking about, is if they're so complex and so well designed, then why would God make bad things so well designed so they could destroy the good? And, and I know maybe it comes down to the problem of evil, but if it's so complex and has changed and adapted to become to actually kill so well, then what's that theological implication?
Right. So that's an excellent question, and I wrestled with this when we first started working on this. The flagellum is a paradigm of beauty. It's an efficient engine, and here we have the same principles now coupled to secreting toxins. And it also looks to be irreducibly complex. In fact, we defined it as an irreducibly complex system because we have the mutants in every single gene required to build this thing, and it knocks out function. If you're going to claim design for the one system, you got to apply it to the other, all right? And it does boil down to this question of theodicy, question of evil. And this would be leveled at me by colleagues as well. Well, how do you explain that? How would a benevolent God allow Yersinia pestis, with this beautiful protein machine gun, to kill 200 million people? But that's a theological question. It's a metaphysical question. It's a problem with the designer. And I think then that you've got to go down the hall to your theologians and ask them, how are you going to explain the problem of evil in this system? Somebody wants us dead, okay? We have, like in Ecclesiastes, our curse is that we have this idea of eternity in our hearts. Yet we live in this, world of death and evil. And I think there are answers for them. I think they're very good answers for them, and that you'll find them consistent in Scripture. And it may be that I look at it as a severe mercy. What's worse than going into eternity in this fallen state? We want, really one of the goals of molecular biology is to make us immortal. We want to understand the aging process. We've got Drosophila now that we grow under certain conditions, certain mutants, and they can have 2 to 3 times their lifespan, normal lifespan. But you want to go through eternity remembering your neighbor's barking dog, And so my mother said, I don't want to live forever and, have that. So it may be, in a sense, a severe mercy. But it gets into a theological question. Why is there death? Okay? And it's a good question.
Yeah, Scott?
Yeah, Art.
I've got a legitimate quick question, but just wanted to say that evidence like this, I think, points to the fact that something is wrong with the current existence, and that if there is this intelligent creator, he would do something to eternally correct that wrong
Right.
somehow, and have a plan
Exactly.
to do that. But I think it's evidence for that. I take issue with a little bit with the phrase, how's it I know it wasn't your term, this omniscient fascist, or what was the...
Supreme fascist.
Yeah, supreme fascist. We think of fascists, I think, as people that are unloving. And
Right,
the
right.
presence of evil seems to me points to, whether it's physical or natural evil or human evil, points to the existence of free will, which must exist if love is to exist. So I would prefer the term, I suppose, supreme lover to supreme fascist, but that's
Okay.
just an opinion.
Okay.
My question is, and I want to compliment you on an outstanding talk. I think constant torque, proton-powered, reversible rotary motor will soon become a household word across the country. You also seem to infer that the concept of irreducible complexity is falsifiable. I was talking to a Nobel laureate here at UCSB a couple days ago, and he said, I don't believe in that irreducible complexity exists. And he did describe himself as something left of an agnostic. His actual words, I'm not making this up. It seemed to fall out of his philosophy rather than his science, but you seem to infer that the concept of irreducible complexity is falsifiable. An excuse for the existence of your flagellum would be that there were pre-adaptive states, that some of the proteins, some of the components of the molecular motor were used for something else. How soon do you think scientists will be able to create, on a gradual, step-by-step basis, the bacterial flagellum in this nanosystem? You know, is it falsifiable? Is it something that one day we'll be able to test in the lab? That's
a good point. You come back to this question of irreducible complexity, and it is so logical. I mean, this is how genetics works. If things were not irreducibly complex, we wouldn't know anything about it, because you'd never get the mutants. You wouldn't disrupt the phenotype. I wish I had a list. If you take out a review article on the bacterial flagellum where it lists every gene that's been identified, it will give the phenotype next to it. Non-modal, at which stage it's blocked. Non-modal, 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 non-able to respond to chemotaxis, non-modal, non-modal. They're all disruptive completely or partially in terms of the function of that system. Most of them are completely disabling if you remove one component. And those genes were identified by the fact that you hit E. coli or salmonella with some mutagen, screened for cells that couldn't swim, mapped all of those mutations, and identified each gene and sequenced it, determined by biochemistry where it was assembled in this organelle. And so how then do we build something with so many interdependent parts in a gradual system? You have to then account for every single protein at some point in its history is having a dual function. It's involved in some other process of the cell and then co-opted for this new function as you're cobbling the system together. So I think that it is falsifiable, and we haven't falsified it yet. Now, there may be systems that are not irreducibly complex, but along with what you're saying, Art, is this idea when I see this subtle change occurring in how we describe these systems. When I was a graduate student, postdoc, everybody was this one-upsmanship on whatever system you were working on, and how beautiful, how elegant, how tremendous, awesome it was in terms of its function. The flagellum is just used as an example. I work on it. Obviously, it's the best example. But people working on ribosomes or DNA polymerase or RNA polymerase would make the same arguments. But then as you follow that thread of logic, then that these are irreducible complex, where do they come from? Now, when I go to meetings, people are talking about Rube Goldberg apparatuses, that these are cobbled together, they're really kind of sloppy. The same people that were saying they were beautiful before are now saying that they're kind of sloppy, because they know what their language is very teleological. in the past, and they want to correct it to fit with the ruling paradigm. So, I see that. I rambled a little bit, but that answered your question.
I guess my question is just to kind of help me understand a little bit better what you're saying, because I got most of it, some of it kind of. Does the proteins that's secreted, the one that's at 30 degrees, it produces the flagellum, and the one at 37 degrees doesn't. Does the one at 30 degrees also produce the dangerous protein that is virulent?
No, they're separate systems.
Okay.
But so the genes for the flagellum are expressed at 30 degrees. Some of them are expressed
So
at
there's
37.
nothing dangerous about the one that's at 30 degrees?
No.
Is it just the mutant in the DNA itself that causes the adverse reaction of creating like the little portal that blows the hole in the white blood cell? Is it just that mutant that creates that, and without that mutant of the gene mutation, that T that was in the middle of it, it wouldn't do that?
Well, that structure is encoded on the virulence plasmid. So all the genes required to build that, what we call a needle-like structure, a little molecular syringe, assemble or activated at 37 degrees. And then when they make contact with a white blood cell, the first protein that's released forms a pore in the target cell, and everybody else goes through it. Okay, so that's all programmed into the system.
So is in the original genes that at this specific temperature to do this, it's not a mutation that
No,
caused it
no,
at this specific temperature?
No. And the point being that the genes that are similar between these two systems are all the ones that are directing at the base of each structure what proteins are allowed to go through. Okay, so that if you had both systems on simultaneously and you're making flagellin or propeller proteins, then they would be faced with this conundrum. Do I go out this system or do I go out this system? If I go out this system, I'm just going to get lost in the environment because I don't have the proteins direct my polymerization into this filament. So that's what's going on. And that, if you're a pathogen, that could be a dangerous thing because they're going to read that signal.
I guess I was thinking just from a theological standpoint, could you say that maybe it was the introduction of something that was abnormal in this system? Maybe the system was originally designed to work in X amount of way, and it was the introduction of something.
Okay, I see your point.
Like,
yeah.
because that could be just the sin factor. If we were to look at this from a biblical standpoint, could we say that, hey, this introduction of this mutation, and all of a sudden everything went screwy, which would follow perfectly in line with saying.
That's really a good point, and a very interesting point, especially as someone that's interested in bacterial pathogenicity, how these organisms cause disease. What I find remarkable, so you're saying essentially that you have this good design, you make a flagellum, allow the cell to swim, find food, reproduce, and that the type 3 secretory structure then is a perversion of that system that now is used for other than its original design. And I think, from a theological standpoint, that is interesting because in bacterial systems, all of the virulent systems that we know, toxins and anti-host factors, appear to have been acquired by some mobile genetic element. This whole structure, which is required to kill, is on a plasmid that can be transferred from one cell to another. Okay, I can cure Yersinia pestis of that virulence plasmid and reintroduce the one from Yersinia enterocolitca and restore full virulence. E. coli 0157, which causes, this horrendous bacterial dysentery that can kill kids by HUS. That toxin's carried on a virus. That E. coli, which is normally part of our normal flora, which we require, it produces vitamins for us, it inhibits other bacteria, acquired a virus that had a toxin on it, and now is a lethal agent. Diphtheria, the same thing. You're not sick from the bacterium, you're sick from the bacterium that caught an infection, a virus infection. And so what I'm saying is that all these virulent systems were somehow acquired by lateral gene transfer. They're out of their original context, perhaps. And now, in this new context, are, until this century, the #1 cause of death in our species.
'Cause it seems like from what the guy was talking about last night, where you change one thing in the structure and then they had to go solve the next problem that caused, and then they had to go solve the next problem that caused, and kind of like it's the same way. And if someone would ask, well, how could a good God create this, blah, blah, blah? Well, it'd be just like, a man that built a car that made it just completely beautiful and complex and told people, don't put sand in the gas tank, don't put sand in the gas tank, and then someone put sand, and then they blamed the designer because the car's running all funky after that. And it's really just, the fact that something was introduced that he hadn't created. And so
Right.
you can't really you can't draw implications of his intent based on something adverse being introduced.
Right.
So,
And that's what you find in our own experience. The technology is always double-edged. We're celebrating the 100th year of, invention of the aircraft from the Wright brothers, and H.G. Wells recognized immediately that this could be a very dangerous weapon, that it would change warfare as we knew it. Suddenly areas that are, at a distance are not so far away anymore. But yeah, perhaps that principle applies. So, good points. Oh,
Hello, I'm a grad student in chemical engineering, actually, and I've noticed that one of the sort of emerging areas of that field is systems biology and studying biological systems from an engineering standpoint. And I know you mentioned something about even in the future it may be required for biology students to take engineering design courses. I was wondering if you had looked any, or if you had been considering through sort of collaborations or anything like that, studying these systems from an optimization standpoint in terms of what ways they might be optimized for the specific functions, and if you could comment on what sort of insights might be yielded by approaching these from an optimization standpoint, from the standpoint of a rigorous engineering design.
I think, imagination's the limit on this. These protein secretory devices, if you're in the pharmaceutical industry as a chemical engineer, then, you want to make pharmaceuticals that are proteins in some single-celled system, whether it's a yeast or bacterium. Always in the past, gram-negative bacteria, which we know the most about, have been a problem because you get contamination with lipopolysaccharide. But here, you could adapt this. In fact, we've actually stripped down the type 3 secretory system so that we just make that needle-like structure, and then we can now design proteins that normally aren't secreted by putting the zip code on them that will direct them to that address and get them out. So from that perspective, it could be very, very important in terms of producing pharmaceuticals. At the same time, these systems are run by plant pathogens, by animal pathogens, by human pathogens. If we can come up with an analog, design a protein that would disrupt this structure from the outside or get taken up and disable it, it would have a profound effect on disease. You take Shigella dysentery, that kills 5 million kids a year in third world countries from dehydration because their food supply or water supply is impure. We've got to understand how these things work, how they assemble, and then we can think about interdiction in terms of how to disable them.
And I'd also like to add just one little comment of my own, and you may wish to add some of this as well. I've noticed among my colleagues in chemical engineering, the assumption is really almost implicit that these things are designed. In fact, many people are studying biological regulatory structures and trying to see what we can learn from them about how to control refineries and plastics plants. And the whole paradigm is, these things are so much better controlled than anything we have ever built. How can we apply this to our own human designs? I haven't heard anybody connect it to intelligent design, but the assumption is just kind of there that these things were designed. And so it's been really kind of amazing to me how widespread that view is.
Yeah, I think it, and it is when you work with them, as you dissect them. You can look at the bacterial flagellum from a biochemist standpoint that Mike Behe does in terms of how these proteins interact. As a geneticist, I look at the program or the algorithm that gets those proteins made in the correct sequence in the right number, and it's another order of magnitude in terms of the complexity. All the feedback mechanisms. That one cartoon that I gave with the steps involved. Those are very precise steps. assemble part A, we want 50 subunits. Check. All right, now we need part B, 35 subunits. Check. You know, is everything kosher? If not, shut down everything below. Start over. And yeah, I mean, they are truly blueprints in terms of the algorithms, and they're very efficient.
One more here, and then we'll take
Okay.
Okay. I hope this will be short. You've been hearing some questions about the problem of extreme, yes, ferocity, even cruelty in nature, the theological problem. But purely from a biological standpoint, I was wondering, it just seems to me like it's not a very good survival strategy for a for a parasite like these bacteria, which you said that they insinuate themselves into white blood cells, take up residence in us. Why do some parasites kill their hosts? It seems like that wouldn't be a very good survival strategy, because if the host dies, so does the parasite.
Right. Well, I mean, yeah, that question is bantered around all the time. And is parasitism a direction of evolution, or commensalism the direction? And we don't know. I mean, that's ascribing a purpose to the system, and supposedly it's undirected, unpurposed. So I don't think Yersinia is thinking, I know, but I mean, you know, this is a bad idea, we're going to kill our host.
But it just seems like that might be, I would think that would be weeded out of its genome if the things that do not work well for promoting the survival and perpetuation of the species would be weeded out.
Right. Well, you've got this tit for tat. I mean, supposedly we're evolving in resistances to these, and you know, an escalating method. But it's a conundrum. Yeah, it doesn't make sense in some respects.